Hello, my name is Elinor Shelp-Peck, and I'm a junior at Meredith College and an undergraduate intern at the National Institute of Environmental Health Sciences. Today, I will present the findings of my internship project that determines the gene expression patterns of human endometrial stromal cells on a single cell level. The uterus has two major compartments, the inner lining endometrium and the outer muscular myometrium. Using a mouse uterus that is structurally similar to a human uterus as an illustration, endometrium consists of stroma, glands, and luminal epithelia, as well as blood vessels and immune cells. During early pregnancy, the endometrium undergoes structural and functional changes to support placenta and embryo development. Stromal cells adjacent to the luminal epithelia change from fibroblasts to epithelial-like morphology, leading to the formation of the decidua. The decidua protects embryos from being attacked by the maternal immune system and provides nutritional support for the developing embryo prior to placenta formation. The transformation process is termed decidualization, which is regulated by the crosstalk between epithelium and stroma cells under the control of pregnancy hormone progesterone. Progesterone acts through the epithelial progesterone receptor to direct production of morphogen Indian hedgehog, or IHH. IHH then interacts with the patch receptor in the stromal fibroblast to activate the COOP-TF2 and EGF pathway to commence decidualization. This process can be modeled in cultured endometrial stromal cells that are isolated from human endometrial specimens, through which we and others have revealed multiple signal pathways pertinent to cell proliferation, immune suppression, angiogenesis, and cell survival. Emerging evidence suggests that human endometrial fibroblasts, or HESF, are primed for decidualization by major transcription regulators prior to hormone-stimulated cell transformation. However, molecular features of HESFs remain largely unknown. To gain insights on this model system, we profiled transcriptomes from 4 HESF cell lines using the single-cell RNA sequencing assay. Dr. Lee conducted the single-cell RNA-seq experiment, and Dr. Wang performed the subsequent bioinformatics and analysis. Of the 4 different cell lines analyzed using single-cell RNA sequencing, 3 samples were primary cell lines, and the 4th sample was of an immortalized cell line. These cells were assayed to determine the number and expression levels of readable genes from a single cell. We profiled 7,054 cells in total with over 1,500 cells per cell line and identified over 6,000 genes per cell. Next, the results from this analysis were graphed based on the composition of the cellular transcriptomatic profiles. The UMAP in figure 2A shows all 7,054 cells from the 4 cell lines after batch effect correction. Here we observed 10 cell clusters numbered from 0 to 9 that are coded with different colors to represent groups of cells that have similar gene expression profiles. I do not have time to show this, but the distribution patterns among these 4 cell lines are well represented in clusters 0 to 6. This indicates that the information extracted from each cluster would reveal common characteristics across the cell lines. These shared molecular features could be subject to further testing in the THESC, which is much easier accessed and handled for subsequent molecular assays. Meanwhile, cells in cluster 7 and 9 are present in specific patients, and cells in cluster 8 are only found in primary cells. These findings implicate the presence of individual differences among human subjects and the unique features of primary HESF. To understand the biological features that are represented by each cluster, we mapped panels of known markers for specific biological events on our dataset to determine their expression levels in each single cell. Figure 2B shows the E2F gene signature, which represents cell proliferation and is relatively high in clusters 4 and 5. The color scale is shown on the right, with yellow being strongly expressed and blue being at lower levels. In contrast, clusters 0, 1, and 3 have high expression of myogenic genes, as shown in figure 2C. This data reveals that HESFs have distinct transcriptomes among unstimulated cells and are not molecularly homogeneous. Next, we identified genes that have 50% higher expression levels in individual clusters over others, as listed in Table 2, and used them as marker genes to further investigate the molecular characteristics of these cell clusters. Genes in cluster 1 failed to meet the cutoff of this investigation, but could be subject to other assays in the future. On each gene in the list, I searched the National Center for Biotechnology Information Gene Reference into Function database for their association with the following keywords of interest: smooth muscle, progenitor cell, stem cell, proliferation, and cell cycle. The results in Table 3 summarize the percentage of genes in individual clusters that are linked to specific keywords. My literature search results echo the gene signature analysis on the cell cycle association. Genes in clusters 4 and 5 have the highest linkage to the cell cycle and proliferation terms, which matches the E2F gene signature mapping in figure 2b. While the vast majority of genes in our search bear smooth muscle association, cluster 2 cells have the lowest association with smooth muscle, showing 100% of their genes linked to progenitor cells. Because previous studies suggest that a subsequent subset of stromal cells retain progenitor or stem cell features, we are in the process of further characterizing the cluster 2 cells. In summary, our data demonstrates that the human stromal fibroblasts are composed of a heterogeneous mixture of subpopulations that allude to the complexity of this model system. Thank you for your time, and I appreciate you watching my presentation.